Thank you, Chairpersons. We thank the ICS for giving us the opportunity to discuss this paper or this discussion. Background, the UTI pathogenesis requires a host response to a urinary tract pathogen such as E. coli. However, urothelial responses are poorly understood primarily because of lack of research in this area. Modulation of urothelial responses may provide new non-antimicrobial approaches in treating recurrent UTIs or complicated UTIs. So let's see. Hit the button. We have previously shown that we can actually dissect a pure urothelium, shown here in the lower right part, from the bladder, and this technique allowed us to be able to patch clamp just the apical cell. You take the urothelial tissue, you lay the apical side down on a glass cover slip using the micropipette. The basal intermediate cells are plucked off, and you can sort of see these hexagonal shaped apical cells, of which you then can come to the basal membrane. This technique was developed by Ming Liu in our lab, who's a first author here. What we detected was a presence of a potassium current, an excised inside-out patch. This is a description of what was in the pipette. And if you go to a symmetric potassium concentration at a 0 millivolt holding potential, you lose current. At hyperpolarizing potentials, you get an inward current. At depolarizing, you get an outward current. We saw two types of potassium currents, a low conductance, 28 picosiemens, and a large conductance, 200 picosiemens. Here are the IV curves. The large conductance was sensitive to calcium in this dose response curve. With increasing calcium on the x-axis, there was more activity of this large conductance potassium current on the y-axis. Now this shows that Paxilene and iberiotoxin, two BK blockers, block the activity of the 200 picosiemen channels. These are the tracings on the left side and the quantification control, and then with the blocker, with abrogation of the BK activity for both using Paxilene and iberiotoxin. mRNA for the alpha subunit of H-slope, which is BK, was found in the urothelium, as shown in this RT-PCR gel. This is probably the most important figure of this talk. LPS significantly increased the activity of the large conductance potassium channel. You see the tracings here before LPS. After LPS, increased activity. Quantification here with almost a threefold increase in activity to LPS. PMB is polymyxin B, which is a blocker of LPS, which reversed the LPS effect. We also looked at the mRNA for putative LPS receptors TLR4, CD14, NB2. These three receptors were only found in the urothelium, but not the lamina propria or detrusor. Here's our housekeeping gene, and the positive control is a spleen. So in conclusion, basal membranes of apical urothelial cells or umbrella cells were patch clamp. We found a large conductance calcium-sensitive potassium channel, likely BK. LPS significantly increased this large conductance potassium channel activity, and the mRNA for LPS receptors were detected in the urothelium, but not the lamina propria or detrusor. Thank you. I look forward to your questions.
Yoshimura in University of Pittsburgh. Nice technical, very nice technique is being used. So what's happening after that the increase of BK conductance in urothelium after LPS? Is it kind of protective against that inflammation? Those type of things can be.
They took away my presentation because I had one more slide because that's going to answer your question because I was anticipating it. But if you can pull up, I referenced 3 papers. We think the apical urothelial cell is behaving like a macrophage. There are several papers, probably 15 years old, that look at macrophage release of cytokines as BK dependent, as the early response. So when you turn on BK, you get a release of cytokines. So we think this is what we're doing next, is looking at cytokine release by the urothelial cells and adding BK blockers to see if we can block the release of cytokines.
Great.
Thanks for that question, Naoki.
One question: if we treat the UTI, treat the E. coli, the BK will be normalized, right?
You mean when you treat it with antibiotics?
Yeah. Yes.
Yeah. Presumably, you would not get the increased activity. I mean, bacteria will cause an increase in the BK activity, so if you have no bacteria there, you won't get that effect, presumably. But I think what I'm trying to say is if we can understand the host response better, we can maybe leverage the host response to not depend on antibiotics, or magnify the host response, or pick a specific host response that we can mimic in an infected host, for example.